it's my pleasure to continue actually what Jay has presented in terms of the work for the next generation tools. And ideally, he would be keeping on presenting it because he's really the brain and his team behind the implementation of this next generation thing. But he's been talking so much that I will try my best to do justice to actually some of the coolest features that come with this next generation tool implementation. So this is what I'm talking about, the next generation tools, which are live. You can go out and try them out right now. I should actually say, if you are used to working with the current implementation, they are going to stay live for foreseeable future. As Jay is showing, we've essentially implemented some features at this point in the new system, and some things might only be available in the old site. There's nothing wrong with keeping on using it, but as we add more and more functionality, we'll not keep on updating the old site, but everything is ultimately going to move to the new site, and we hope we'll convince you that it's going to make your life much easier. So to just recap, that's where Jay left us. We had these input sequences here from dengue 1 through 4 envelope proteins, and done binding, processing, and immunogenicity predictions for it. So that was entering the sequences, choosing the alleles, and running, again, this is just what Jay did before, adding these other prediction methods to get like processing and immunogenicity in there, running it, and then you have this output table. And now this is a more complete output table, and it goes a long way. And actually, if you there might be more columns to the right, you're going to have to scroll to the bottom of the table to actually scroll left and right to see them all. And you have a lot of rows here, but you have to watch here also at the top. Let me actually change to a laser pointer. There's more tables. There's the sequence table. So that echoes your input. These are to remind you, like these 1 through 4 is the index of the sequence. So that's what's echoed here. And then you also have the net MHC pan allele distance, which gives you an idea of the closeness of that allele in the net MHC pan algorithm to the neighbor it uses to predict. Actually, in this case, these are all standard alleles. There's nothing special to display there. So in this peptide table, which is the default output, you have all these columns that we looked at quite a few times, and you can use this table to filter these further. So you can go in here and say, I just want to see the ones that have a predicted medium binding percentile from the consensus output method that is 2 or lower. You click, you might also say that your processing might be in a certain range, and that your immunogenicity might be in a certain range, so both of these above 1. And then when you do this, these are the standard kind of filters that you might be familiar with from Excel or from Google Sheets. And then you have now limited this to 50 of the 276 rows, and you can save that table state, and that now is no longer just like on your browser, but this is now the output of these predictions, including these filters being set. Now, these results you can pipe on to the next tool, because we found that this is what you commonly do when you predict. You start with one set of predictions that you're making, and then you want to do the next and the next, and we want to kind of maintain this stepwise approach to selecting ultimately a set of peptides. So to do this pipe building, you will have seen on the top left of the tool. So this is what Jay was showing primarily here, these elements. On the top left, you have the pipeline map, which if you just have the T-cell class 1 predictions, that's essentially one step in the pipeline. But you can now add here to send the results of this to cluster, to the cluster tool, and then you have one or more selections what you're going to send. Here it says peptide, that corresponds to the column, the peptide column in here. And essentially you can only select columns, obviously, that are useful. So we're not doing clustering of whatever peptide lengths. The clustering tool is taking peptide sequences and input. So these peptides now, the 50 out of the 276 that fulfill your prediction criteria can be sent on in this piping format to the cluster tool. So when you do this, now it's going to ask you how to run the cluster tool. the cluster tool essentially has a few input parameter, namely, so it's trying to identify within your set of peptide sequences which ones of those are similar, and then you have essentially defining here what is the minimum similarity that you want to have peptides to be included in the cluster for. You can set peptide lengths, minimums or maximums, or in our case, we will not. There's other ways of how to build a cluster. There's a paper for this that is linked in the method. How to define the clusters. You can say all connected peptides get put in a cluster, or only clicks, namely fully connected clusters, or you can do our cluster break method, which we would recommend, and then run it. So this now takes these 50 peptides, and you're getting, first of all, some warnings. And these are warnings that are specific to the cluster tool, which actually, when you click on them, tell you that there's a number of duplicates in there in the input sequence. And that's actually kind of expected, because we were using 4 dengue envelope sequences from related strains. So you can imagine that the regions that are conserved across the strains are going to occur multiple times. And by the way the prediction tools works, is I'm then going to identify these as being duplicates. But in the clustering, it doesn't know how to deal with duplicates, so it's just going to remove them. So now you have then the cluster results, which are going to appear in the similar peptide table, similar to what you've been seeing now several times in the previous presentation, which are identifying. So say here for cluster 2.1, you have peptide 1, 2, 3, 4, 5, 6, and 7 in it, and then the consensus. And you can see here the aligned peptide, KQWF, etc., RQWF, and you see here the alignment with the hyphens indicating why they are put in the same cluster. And then you have here the consensus sequence of these, where residues in the consensus that are not clear are replaced by an X. So these are then, as you can see here, these are clearly overlapping peptide regions, and you don't want often to synthesize, say, multiple peptides that are essentially the same, and rather work with the consensus. So you can interact with this table the same way like the other tables. So you can sort, or you can filter. So you can also do the same kind of saving, downloading, resetting these, set the display comps you want to see, etc. It's the same functionality. We're going to use the same kind of table library here throughout the site. So, oh yeah, and I should point out here, up here, so you have the peptide table, and the cluster tool also has visualization as a separate option to look at these results. So this is a table, and this is showing the interconnected map of each peptides. And you can actually mouse over, or you can select them here and see like the consensus sequence of the cluster and the individual peptides in it, to just visualize what you have here. So you can, as I said, we can filter this, and you don't have to filter by numbers. In this case, as I showed you had for each cluster, you have the identification of like peptide 1, 2, 3, 4, 5, and then something is called one entry is called consensus, which is not really one of the peptides that was given there, but it's a new record. So you can filter by consensus, and now you're going to get the 32 rows that are corresponding to cluster consensus sequences, and you have them here. So you really have a new sequence compared to what was your input data. So if you have done this kind of filter, as always, you must save stable state, table state before moving forward. This is kind of like, you know, we're looking at this pipeline, and you might just want to sort one way or the other to look at the results or filter them. There's a difference between how you look at it and how you want to send the results forward to the next step in the pipeline. So you're going to have to save this table state, and then we can add the next tool, which is going to be the PEPMatch tool. So those are the 3 that we have available. And this PEPMatch tool here then has 2 possible inputs. You could use the peptide column or the cluster consensus column. And just because this is more amazing, we're choosing cluster consensus here. So ultimately what you're doing now is we've started with a set of dengue envelope peptide sequences for a representative of all the different serotypes. We have identified likely binding peptides in there in a vast set of MHC class 1 alleles. that are also likely to be processed and immunogenic, then we've sent them to the clustering tool to identify these 32 essentially regions in there that have like these overlapping peptides that are there. And now we're going to look in the PEPMatch tool. So we choose the cluster consensus, the consensus sequence for each cluster, and that is now piped to the PEPMatch tool. So when we click this, we again get the parameter for running that tool, which is now a PEPMatch is essentially looking up for a given peptide sequence, and that can include an X. What are the best hits in a given reference proteome? So the typical application here is for the host proteome that you're looking at. So we're looking for dengue virus envelope proteins that are peptide regions that are potentially found conserved in the human proteome, which might then be a sign for possible autoimmunity. So we're including the best match for each of the peptides that is the input, the peptides here being the consensus sequences. We're expecting, so that's the two options. We can also do like, we can limit it to, it allows up to 3. We run it, we select human, and then we get this output, where you now have for the input sequences, which were the clusters, you get the match sequences and where do they match in the human proteome. So you can again interact with this table the same way like you're used to. You can save, display, all the same things as before. I just want to end here for a bit. Like this is obviously one of many possible applications, and as we are adding to these pipeline tools, the ability to customize this for your purposes is just going to continue to grow. Sorry, where was I? Okay, I don't see the link here, but on the top of the page, there is the link to the help in general. So if you have questions on how the cluster tool works or the pipeline tool works, you can access this help here. And that's also one of the, and actually help about the pipelines themselves. Previously, we had all these tools living by themselves, and it was hard to like provide one consistent documentation. Now it's on here. This is the cluster. This is for PEPMatch, and this is the timeline for release. Jay already said this. I'm surprised, Nina, that I thought I had another 5 minutes, but I guess I'll stop here because Jay has already gone over it. I think I should point out, and I did say this, like the legacy will eventually be migrated, but they are still there for the foreseeable future. And that's it. Nina?